Welcome to fast electrophoretic analysis of genome libraries in the next-generation sequencing workflow. I'm Timlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. Over the last few years, novel sequencing platforms have greatly lowered the cost per base of DNA and RNA sequencing. In particular, lower costs coupled with significant improvements in data quality have enabled sequencing projects that may have previously been too expensive to complete. As a result, the demand for high-quality libraries has escalated. Laboratories have invested significant effort in improving library preparation and workflows through introducing new equipment and automation. In particular, the need for quality control technology that is compatible with established workflows and in-place next-gen sequencing instrumentation has assumed increasing importance. The speakers today, Paolo Piazza and Donna McDade-Walker, will talk about their experience and successes incorporating new equipment into their lab flows in just a minute. Before we get started, though, I want to encourage you to submit questions for our panelists. We'll answer as many questions as we can after the final presentation. Simply type your question into the Ask a Question box and hit Submit. Donna McDade-Walker, Senior Product Manager and Marketing Team Lead at Agilent Technologies, will kick off the webinar today with a discussion of a new solution that can QC fragmented DNA, whole genome libraries, and target enriched libraries, specifically the Agilent 2200 TapeStation system. Donna, we're ready when you are.
Thank you, Tamlin. Hi, everybody. Thank you for attending this educational webinar hosted by GEN. I'm Donna McDade-Walker, and I'm joined today by Dr. Paolo Piazza from the Wellcome Trust Centre for Human Genetics in Oxford. And today we'll talk about fast electrophoretic sample analysis in the next-generation sequencing workflow. Next slide. So this slide is a summary of the webinar today. I will start with an introduction to the next-generation sequencing workflow, and I'll illustrate the role of the current gold standard for sample QC, the Agilent 2100 Bioanalyzer. Then I will introduce the new system from Agilent for next-generation sequencing sample QC. That's the 2200 TapeStation system. This will be followed by a presentation from Dr. Piazza about their experiences with the TapeStation and how they've implemented that system within their everyday workflow. And then lastly, I will present a comparison study of the Bioanalyzer and the TapeStation within the next generation sequencing workflow, in particular the Illumina workflow with the SureSelect target enrichment and focusing on DNA samples. Next slide. So I will now talk to you about next-generation sequencing and the Agilent 2100 Bioanalyzer. Next slide. This slide, the next-generation sequencing workflow, is a very broad overview of the workflow itself. First, you extract and you purify your native or synthesized double-stranded DNA. And then, because it's so large and you want to work with it, you need to fragment it. After attaching your adapters, you then amplify your library. And at this stage, you can choose whether to enrich your target or not. So this particular step, this enrichment step, is used to enhance your area of interest, whether it be analyzing the exome or whether you just want to focus on a small targeted panel. The next stage, it's a further amplification step, and this is before continuing on to the sequencing step. So some of the sequences, they need you to amplify your sample using cluster generation, so that's bridge amplification, or for other of the sequences, they want you to do some emulsion PCR to amplify your library before you then move on to the sequencing itself and all the data analysis that goes along with that. So my next slide is a simplified workflow for DNA library prep for the Illumina. Two stages that you can see highlighted with the Bioanalyzer chips, these blue DNA Bioanalyzer chips, indicate that this is a step that should be followed by sample QC in order for you to monitor the size distribution and for the sequencing library, the size range, the purity level, and the quantity of your library. This is really important as a library preparation artifacts such as primerdimers or chimeric inserts or adaptodimers, they can be sequenced together with the intended template and therefore reduce the number of meaningful sequencing reads. So really, identifying artifacts at this stage allows their removal and it avoids wasting sequencing capacity. In addition, the Bioanalyzer allows you to quantify the contaminating products as well as the libraries, so this enables the calculation of the library purity thresholds. The next slide shows the Bioanalyzer system and a little bit of the history that goes along with it. It was launched in 1999 and it can analyze DNA, RNA and protein. It also has the industry standard for the analysis of RNA, so that's the RIN value. And it's also considered the gold standard for next-generation sequencing sample QC. And to back that up, really, we've got, since its launch in 1999, it has amassed more than 15,000 citations in peer-reviewed journals. The next slide. So for the next part of the talk, I will now move on to the new Agilent 2200 TapeStation. So my next slide. The Bioanalyzer and the TapeStation, both from Agilent, are very similar products in that they perform electrophoresis and analysis of DNA, RNA, and protein samples. In addition, the TapeStation is able to offer the ease of use and scalable throughput, meaning that you're able to run 2 or 3 samples on one day and then perhaps 96 samples the next. And the TapeStation is able to accommodate that without compromising the cost of your experiment. In addition, this scalability follows through to the time to result. So running 2 or 3 samples will take approximately 5 minutes, whereas running 96 samples will take perhaps an hour and a half. Overall, your results usually take approximately 1 minute per sample. Next slide. So you can see on this slide that the overall concept of the TapeStation system follows this 1, 2, 3 approach. We have a consumable called screen tape that carries out the sample electrophoresis, which is placed into the instrument, the TapeStation, and the TapeStation loads and separates the sample and the results are analyzed with a software package. This slide also illustrates the ease of use of the system. You remove the screen tape from its foil packaging. It's completely pre-packaged and ready to go. You don't need to do anything to it. You then place the screen tape into the TapeStation with filtered tips, and you put your samples into the TapeStation and you press start. Your results are then ready within about 1 minute per sample. So my next slide, the Agilent screen tape slide, shows our current range of screen tape, and you can see that we offer DNA, RNA, and protein analysis. Each screen tape is made up of 16 individual separation channels, which allows for the scalable throughput, as well as eliminating any sample carryover. We have DNA and RNA in standard and high sensitivities, and the DNA screen tape displays the same sensitivity as the Bioanalyzer high sensitivity kit. We also offer an RNA Quality Metric that has been validated against the Bioanalyzer RIN, which is called the RIN-E, or RIN equivalent. The TapeStation instrument itself is on the next slide. And this shows that the TapeStation will load your sample onto the screen tape using individual loading tips and then applies a current to the screen tape. The TapeStation can hold either 96 well plates or 8-way tube strips and has an integrated barcode reader that automatically recognizes the screen tape type and loads up the required parameters for running it. So it knows how much it needs to load, it knows how long it needs to electrophoresis for, it knows exactly which analyte you're going to load onto that screen tape just through the barcode. This is also how we track previous usage. So if you have run 2 or 3 samples on a screen tape on a previous occasion, you can just place it into the fridge and you've got 2 weeks to use the remainder of the tape. Once you then place that used screen tape back into the TapeStation, the barcode is able to tell the software that 2 or 3 of those lanes have already been used, and it will then just load your fresh sample into the next available lanes. We also have 2 instruments available. We have a nucleic acid only instrument, and we also have a second instrument which can analyze DNA, RNA, and proteins. The next slide shows our analysis software. Now, this software has a real familiar look and feel, as we've based it on the Microsoft Ribbon concept, which you can see at the top here, and it uses the same nomenclature as the Bioanalyzer Expert software. So all of the buttons have a real similarity to them, so that anybody who's familiar with the Expert software will easily be able to integrate with the TapeStation software. Down the left-hand side here, we have a navigation pane for the easy selection and identification of samples. And you can also see that we have a gel view. Towards the right-hand side, we have an electrophoregram. And at the bottom there, we have our data table. And that can tell you everything from the molecular weight, the concentration, percentage of integrated sample, distance down the track. These can all be chosen within our column chooser and set up for the next time that you want to use it. Specifically for next-generation sequencing DNA sample QC, you can see on the next slide, we also have a region view to present the average size, the concentration, and the molarity of samples. Now, this is exactly the same as the Bioanalyzer Expert software, which also offers this function. We also have a very simple reporting tool that allows you to export the data from the region tables to a CSV file, to Word, or to a PDF. For total RNA QC, you can see on the next slide, the 28S and 18S are automatically detected and labeled, and we have optional RNA color coding that can be easily set up by the user for the really easy identification of sample quality. So you can color code a particular RNA range green. If you wanted that to signify your good RNA, you could set an amber color for medium RNA, and you could set a red color for poor RNA. The next slide shows the Agilent screen tape RNA Quality Metric, the RIN equivalent, or RIN-E, has been validated against the RIN and returns a RIN-E value with a median error of less than plus or minus 0.4 RIN units, which is an excellent correlation and shows the applicability of the RIN-E for standardizing RNA quality within the next-generation sequencing workflow. So I will now hand you over to Tamlin.
Thanks, Donna. Before we go on to our next speaker, I just want to remind everyone that you can submit questions at any time by typing a question into the Ask a Question box on the lower left of your screen and hitting Submit. Our second speaker, Paolo Piazza, is a postdoctoral research scientist at the Wellcome Trust Centre for Human Genetics. He'll describe his lab's next-generation sequencing workflow and how the Agilent 2200 TapeStation fits in and provides qualitative information. Paolo, you can begin.
Thanks, Tamlin. So I would like to spend a couple of words about the Oxygenomics Center before starting. The center itself is located within the Wellcome Trust Centre for Human Genetics in Oxford. And next slide. It's one of the 4 MRC sequencing hubs in the country, and is the result of further restructuring of the existing core facilities. And the aim was to set up a center that was able to face the challenges of genomics research. Next slide. The center itself takes advantage of the synergistic cooperation of the HalfRobot Genomics Center and the Bioinformatics and Statistic Genetics Group. So together we have a setup that is able to cope with the majority of projects or adapt ourselves to accommodate for new projects that we have not dealt with before. Next slide. Concerning the sequencing capacity of the center itself, we made a strategic decision to focus on Illumina technology, and we utilizes 5 HiSeq 2000 and 2 MySeq. This was recognized as the technology that would support the majority of the projects and all our needs for the foreseeable future. Next slide. So it is commitment of the center to be able to support as many application and methods as necessary without compromising on quality or turnaround times. So this slide shows a breakdown of the 3 major applications, so gene expression and regulation, genotyping, and epigenetics. And also there's a breakdown of all the libraries that have been constructed since 2011. So as you can see, even within one application, there's a number of different protocols that can be adopted, and we have thorough conversation with the users or collaborators to decide and advise what the best solution for their project. And in the next slide, here is a schematic representation of the workflow for the library construction for genomic DNA. And as you can see, it's divided into 3 main parts. We have a pre-PCR, post-PCR, and sequencing dedicated environments. This is meant to minimize the possibility of cross-contaminations between samples. And the next slide, if you look carefully at each and every step of the workflow itself, you can see that sample QC, fragmentation, and repair, A-tailing, ligation, and so on, are all steps that are amenable for some sort of QC. And all these steps are normally QC'd if it's a new protocol that is to be evaluated in the center or in the group, and this is a good practice for any group that wants to start up to deal with these technologies. However, there are, in the next slide, certain steps that is always good practice to QC, particularly the sample as they are about to enter the workflow need to be QC'd. This is obvious because this is a starting point of long and potentially expensive procedure, so you want to make sure that everything is in order since the very start. In the case of genomic DNA, when you fragment, you want to make sure that the fragmentation, the population of fragments, is going to represent the sample. Equally important is to QC the libraries themselves at the end of the process, since the sequencing is then going to be the most expensive part of the whole workflow. So you want to make sure that the libraries that you're going to sequence are of good enough quality. And next slide. I would also like to remind everyone how big an impact the high throughput sequencing technologies have had in the field of genomics. So this is a slide showing the implementation of Sanger sequencing for the Human Genome Project and how important has been the automation and the implementation of the capillary sequencers. And this technology, next slide, survived for over a decade. And despite the fact that it gave the great achievement of the Human Genome Project, it wouldn't be adequate for the current projects that are supported by HalfRobot Sequencing. What is remarkable about the new sequencing technologies is that they exploded, so to speak, very rapidly, and they keep on improving, and new technologies keep on coming up. So that where we were talking about decades before, we're now seeing improvements and changes on a yearly basis. For these reasons, next slide, it is very important for a setup like ours, but also smaller labs, to surround ourselves with a number of equipment that allow a rapid adjustment to the newer technologies or newer applications within for a specific technology. So this is an example of instruments that can be used for quantitating DNA. Next slide. Given the increased throughput of the sequencing platforms, an increased number of samples can be processed and has to be processed. So the implementation of liquid handling is also important. And finally, next slide. There are additional instruments that can be used at different steps throughout the workflow to facilitate the work of the lab. I will now focus on one instrument in particular, which is in the middle of the slide, and it is the TapeStation. We've been using this instrument for a very long time, as you can probably judge by the fact that the colors here are different from what Donald has shown before, and this is because we implemented this instrument when it was still a Lab 901 instrument before being Agilent. And the decision was made because at the time we were looking for something that, similarly to the Bioanalyzer, would provide us with good quality QC data, but we needed something that was going to be more easy to use, flexible, and scalable. And the TapeStation was the instrument that best suited us at the time and still does. So in the next slide, so this is an example of an RNA sample QC, and there's a specific tape that allows us to do that on a TapeStation. As you can see, there are two samples here. The one on the left-hand side has a nice profile showing high level of integrity, and the RIN value equivalent is also high. On the contrary, the samples on the right-hand side have obvious signs of degradation, and the RIN value is much lower. So having a number to quantify the integrity of a sample is extremely useful because it allows us to determine threshold over which we will fail a sample. Integrity of RNA is very important for RNA-seq since the enrichment of mRNA itself is based on poly-A. We need the fragments of RNA to be intact during this process and before sequencing. Degraded RNA will lead to artifacts and missing transcripts or region of the transcript. In the next slide, we also deal with a fair number of chromatin immunoprecipitated samples, and this is a very quick reminder of how chip works. So from fixed chromatin, the sample gets fragmented and then captured using specific antibodies for the protein of interest. And both the fragmentation and the immunoprecipitation steps are crucial to the quality of the sample. So we want to make sure before starting the prep that the size distribution and the amount of sample that we receive are enough or good enough quality. In the next slide. So we QC chip samples using the high sensitivity version of the D1K screen tape. And again, you can see on the left-hand side a nice profile of a fragmented chip sample, whereas the sample on the right was a fail because it wasn't detectable. And like in any other case where we fail a sample, we always go back to the customer or the collaborator and discuss alternative solution for their needs. And finally, in the next slide, is an example of a QC of genomic DNA, where throughput is not necessarily a big problem because, as you can see, we can use precast gel or a high-density type. Assessing the quality is very subjective, and this is something that we really would like to move away from. So as an example, on the right-hand side, there are 3 lanes. The first one with the path shows very nice intact band of genomic DNA. The one in the middle shows clear evidence of the presence of RNA contamination, which may interfere with the process. And the third lane shows evidence of degradation of the sample itself. And in this case, it's fairly easy to fail those 2 samples because the degradation is obvious and the contamination is obvious. But in cases where we have partial degradations, it's really subjective, and it's all down to experience to determine whether to pass or fail a sample. So having an instrument like the TapeStation that analogously to what it can do for RNA could quantify the integrity of DNA is something that the field really needs, and definitely we do need something like that. In the next slide, I want to refresh the memory about how the Illumina sequencing technology works before going into the details of QC in the library preparation steps. So at the beginning we have some sort of fragmented DNA, whichever origin that comes from, whether it's a chip or a genomic fragmented DNA or RNA. It's ligated to specific adapters. Next slide. and the ligated product can now be denatured into single strands. Next slide. Which can then hybridize onto the reverse complement of the oligos immobilized on a flow cell. And those priming sites allow for the synthesis of the complementary strand. So next slide. This allows for, next slide, for the library itself to be attached to the flow cell very simply. And moreover, this product is now capable, usable for amplification. So in the next slide, you can see how the DNA is free to bend over to other oligos on the flow cell, which can then prime the synthesis of an additional strand. And if you do that for a number of cycles, in the next slide, you can see the formation of a number of clusters. Each cluster is originated from a single molecule that was initially flowed over the flow cell. So having said so, in the next slide is an example of a fragmented genomic DNA. And what we want to make sure here is that we have the size distribution of fragments that is compatible with the technology we want to use and the application we want to implement. It's important to note that if the fragments are too large, that bridge amplification becomes very inefficient, and the clusters themselves can interact with another. So this is a crucial step to make sure that we have a good starting material for the library construction. In the next slide is an example of different steps at which we can QC library construction for a shoe select protocol. And after the fragmentation itself, we check the pre-capture library, so before going into the hybridization with the specific probes, and also after that same capture. In what we want to see is a gradual increase of the peak size of the library. This is because of a combination of the ligation of adapters, but also because of the serial cleanups that occur during the protocol. And in the next slide, as I mentioned earlier, we produce wide variety of library types. And the TapeStation allows us to QC most of them on a single type of tape, which is very convenient. But also you can see here that we have, on the top left, we have a library with high insert size and very tight size distribution, compared to the one next to it, which has a broader size distribution. And then again, at the bottom, there's an example of a shoe select library. So different libraries can all be QC'd very easily on the same tape. But apart from determining the size distribution and peak size, we also can look for artifacts. So in the next slide is an example of adapter-adapter peak, which is clearly identifiable. And this is something that we want to prevent or eliminate before sequencing, because those small fragments don't contain any of the original sample. They're just empty adapters, so to speak, but they're still capable of generating clusters and being sequenced. So, in fact, they deplete the sequencing results from usable data. In the next slide, I'd like to spend a couple of words warning people that no matter how much you automate and how much thoroughly you QC your things, there's always room to learn and there's always room to use your brain. These are two examples that I'm going to describe now. The first one here is from a library produced in Nextera kit from Amplicons. And from the profile obtained with the TapeStation, we noticed that there was a potential long tail, which I hope you can appreciate, towards the upper marker. And we confirmed that by, in the next slide, running the same library onto a Bioanalyzer chip, which allows for larger fragments to be visualized. And as you can see, the size distribution is quite vast, going from about 150 to almost 10,000 base pairs. So this is obviously not a library that we would normally like to see or to sequence. So this would have been a fail. But despite different attempts to optimize the protocol, we always obtain the same profile, suspecting that this could be some sort of artifact within the nature of the library itself. So we decided to sequence the library nevertheless, and we obtained very good data. And interestingly, the profile and the size distribution and the peak size obtained by the sequencing data showed that the peak was around 144 bases. So based on this experience, we now adjusted our threshold for QC in those specific libraries, take this data into account. The next example shows what appears to be like a very nice library within range and with a good intensity. However, after sequencing, in the next slide, we noticed that the library itself was very low complex. So the panel on the right-hand side is an IGV screenshot showing two libraries, the one on the top being the one that I'm discussing now, and the one at the bottom, a previously sequenced library. So the one at the bottom shows what the sequence reads should look like. Every line is a read mapping onto the genome. As you can see, the top sample shows huge gaps where there's no representation of the genome at all. And this is correlated with poor complexity of the library itself. So we now introduced additional QC steps to make sure that the libraries are going to be providing good quality data at the end. In the next slide, as it often happens when you have an instrument that works well and you like, you want to use it as much as possible, and you start thinking of other ways of using it outside the purpose of what it was bought for. And in this case, particularly, we wanted to quickly assess whether a method for mRNA enrichment was working well in our hands. So the sample on the left is the total RNA that we started with, showing the two ribosomal peaks, and the same sample after treatment with the RiboZero kit on the right-hand side shows that the ribosomal peaks are almost not identifiable. So we started the prep with high confidence that the sequencing was going to be good, and the results was, in fact, as expected, of very good quality. The next slide is an example of another ways we want to use the TapeStation for, and it is to calculate relative quantities of new libraries compared to previously sequenced ones. So the aim of this is to be able to quickly determine the amount of library that we need to use for the sequencing. And in this case, we run libraries that we sequenced in the past with new libraries that we were about to sequence. And comparing the molarity calculated by the software, as you can see highlighted in red on the bottom panel, we were able to calculate the concentration to be used for the new libraries. And in the next slide is the results after sequencing. So the red line shows what we were aiming for. We were aiming for 800,000 clusters per millimeter square. And as you can see, all the libraries placed themselves very close to that figure. So this was a very remarkable result for us as being a pilot, and we are now looking to do more experiments to see whether we can implement this into production. We suspect that, as for many other ways of determining the concentration of libraries, it's very important that the comparison is done with a library as similar as possible in terms of size distribution, intensity, and possibly GC content. And the next slide is a final example of what we were able to do with using the TapeStation. In this case, we were QCing the libraries before sequencing, and we noticed the presence of the adapter-adapter peak, as you can see in the panel, as a lower molecular weight peak. As I said before, this is something that you don't want to have in your library, so we wanted to do an additional size selection step And this was a group of 4 libraries that we needed to pull together for the sequencing. So because we were running close to the end of the project in terms of deadlines, we decided to use the TapeStation to calculate the concentration of each library only for the portion that was of interest, so excluding the peak of adapter. Use that concentration figure to pull the libraries equimodally, run a single lane on a gel, and quickly site select, so saving quite a vast amount of time. And in the next slide is the result of the pooling after sequencing. And as you can see, they're all very close together in terms of being equimodally represented, with the exception of the 4th sample, which is slightly overrepresented, but still within acceptable range, and every sample got enough data for the project. So all in all, this was a success. So I think I'm done here, so I can give the line back to Tamlin. Thank you.
Thanks, Paolo. Both of our speakers will be available to answer questions after Donna's conclusion, so I encourage you to submit them now. Donna will join us again to compare results from various stages of the Agilent SureSelect Target Enrichment Illumina NGS protocol to the current gold standard, the 2100 Bioanalyzer system. Donna, we're ready for you again.
Thanks, Tamlin. I will now present a comparison study of the current gold standard, the Bioanalyzer, with the TapeStation within the NextGeneration sequencing and Agilent SureSelect Target Enrichment workflow. Next slide. In this slide, I've presented a high-level flow diagram of a DNA sample as it moves from extraction to sequencing. During this workflow, it's important to analyze the size distribution, purity, and concentration of the sample to aid successful sequencing. As a minimum, the following 3 QC steps have been identified, which I have highlighted in pink. So you can see we have the genomic DNA extraction and fragmentation, and then you perform a QC step. And you really want to have a look at your size distribution at this stage. You then do your end repair, your adapter ligation, and your PCR enrichment, and then you do QC2. This, you're particularly looking at the average size of your DNA smear, and you want to know your concentration and purity. After the target enrichment step, which we performed within this experimental study, you then amplify again and you do your QC3. Again, you want to have a look at the average DNA smear size, and you want to have a look at the concentration and the purity of your sample. After that QC step, you then move on to your next generation sequencing. Our next slide is the experimental study, and it gives you an overview of the work that was carried out by the SureSelect team in Santa Clara. They obtained genomic DNA from breast cancer and skin cancer tissue, and then they followed the SureSelect Illumina workflow, analyzing the samples at QC1, QC2, and QC3. In addition, 9 exome library samples were sent to us by a customer, so they'd already been processed through QC stages 1 and 2, and they were analyzed by us on the Bioanalyzer and on the TapeStation. All samples were analyzed once with the current gold standard, the 2100 Bioanalyzer, and in duplicates with the 2200 TapeStation. So this next slide, QC1, gives you an overview of the results that we obtained. So in each of the graphs, the Bioanalyzer is the blue bars and the TapeStation is the pink bars. The top graph is a comparison of the values obtained in nanograms per microliter using the screen tape D1K on the TapeStation and the Bioanalyzer DNA 1000 kit. The TapeStation values also show the standard error on each bar, although in some cases it is a bit difficult to see as they are so small. So for instance, in the top graph, SC3, so that's skin cancer sample 3 and skin cancer sample 4, they have very small error bars, but they are present. Within the graph itself, you've got BC1 to BC4, that corresponds to the 4 breast cancer samples, and you've got SC1 to SC4, which corresponds to the skin cancer samples. The overall deviation for the reported concentration values at QC1 step was less than 10% between the two systems, and the average sizes obtained, which you can see in the lower graph, were calculated using the region views, and they showed a correlation within about 5%. In the next slide, we have QC2, and you can see that the results are very similar. We have a deviation of 10% for concentration values in the top graph, and approximately 5% for sizing values using the Bioanalyzer DNA 1000 kit compared to the TapeStation D1K screen tape. The next slide is the QC3 results, and you can see that we used the Bioanalyzer high sensitivity DNA assay and the TapeStation high sensitivity D1K screen tape, and we have a correlation of within 12% in the concentration values and less than 5% deviation in the sizing values. The next slide also shows the QC3, and this is the customer data from exome libraries. So the size and information and the molarity obtained from the Bioanalyzer is usually used to dilute the samples to the required concentration for clustering at this stage. So what we did was we analyzed the samples and looked at the molarity of each sample compared on each of the high sensitivity kits. And you can see that we had an overall deviation in the reported molarity of less than 10%. Within next-generation sequencing workflow, this is a critical step, as it's the last opportunity for checking you have a good quality sample before moving on to, in this particular case, cluster amplification. In the next slide, so we have shown here that the performance of the TapeStation system is comparable to the current sample QC gold standard, the Bioanalyzer. The new 2200 TapeStation is capable of providing excellent qualitative information about samples, enabling informed decision-making at each stage of the next-generation sequencing workflow, and it has the added advantage of scalable throughput, ease of use, and a fast time to result. So moving on to the next slide. We have shown that the Agilent 2200 TapeStation system is an excellent tool for next-generation sequencing sample QC that can be easily implemented into the workflow and allows the flexibility required for the majority of next-generation sequencing laboratories. In the next slide, my penultimate slide shows that if you would like more information about the TapeStation system, for instance, application notes or information about each of our screen tape ranges, then please do visit our website. It's www.agilent.com/genomics/TapeStation. And on this final slide, I would just like to acknowledge all the people from the Oxygenomics Center that were involved in generating the data for Dr. Paolo Piazza's presentation. Thank you very much. And that's back to you, Tamlin.
Thanks, Donna. I have a lot of really good questions for you, but before we start the Q&A, I just want to remind everyone one final time to submit questions at any time. All right, so let's get started with our Q&A. Donna, the first question we have for you is, what was the impetus for changing the RIN algorithm?
The RIN value is an algorithm that was developed using the RNA traces generated from the Bioanalyzer and their lab chips. So it's so specific to the Bioanalyzer system that it can't really be transferred to other systems. So the RIN value is still the gold standard for RNA QC, and it remains with the Bioanalyzer and the lab chips. But what we've shown is that the TapeStation system correlates excellently with this RIN value, and hence the term we've used, the RIN-E term, or the RIN equivalent.
All right, Donna, how long will it take for 96 samples to run?
It takes about an hour and 30 minutes. We compensate our electrophoresis times to the temperature of the instrument. So the warmer the matrix, the faster your analyte will move through it. So the upshot is a warmer instrument will take a shorter amount of time, but on average, we say round about an hour and 30 to an hour and 40 minutes for 96 samples.
Okay, Donna, this listener says, I've heard that capillary electrophoresis systems can suffer from sample evaporation when processing as many as 96 samples. How does the TapeStation counteract that?
Right. Well, other systems, they can have some delicate mechanical parts that are moving the samples around within the instrument, but we just use the individual filtered tips. So we can apply a foil seal onto the top of our 96-well plate, and then the tip will just pierce through the top of that and lift the sample from underneath. So we've validated the performance of DNA, RNA, and protein on the TapeStation for the length of time it takes to process that whole plate, and we don't see any effects with evaporation. The performance stays the same.
Donna, how easy is it to swap between the different screen tapes, for instance, from DNA to RNA samples?
It's really, really simple. It's literally just removing the DNA screen tape and replacing it with the RNA screen tape that you've just literally taken out of a foil packet and you've placed it straight into the instrument. And then you quickly swap out your samples and you're good to go. It's a really flexible system for jumping between any of the samples, actually, from DNA, RNA, and protein and back again.
All right, Donna, can the TapeStation be used to quantify and assess the quality of genomic DNA unfragmented?
At the moment, currently, no. The highest DNA, the largest DNA fragment that we currently analyze is up to 1,000 base pairs. However, we do have an ongoing project that's very close to completion that will be able to run genomic DNA, and it will be able to provide information on sizing, on the concentration, and the integrity of that sample. So watch this space.
All right, thanks, Donna. Paolo, I have some questions for you. Specifically, someone wants to know, are these genomic DNA data from TapeStation?
No, as Donna just commented, there's no possibility at present to do it on the TapeStation, unfortunately. So they were all done on gels.
All right, and then the listener says, I'm not sure if I fully understand the scalability and constant cost comments. Could you please explain further?
So one of the reasons why we decided to buy the TapeStation almost 2 years ago now was because we wanted to be able to have the freedom of run just a couple of samples without having to pay for the whole cartridge, as it would be the case for a Bioanalyzer, for example. So what the TapeStation does is that you have your cartridge which you load, and then you will only use the lanes that are required for the number of samples you need to run, which means that the rest of the tape remains sealed. You just put it back in the fridge and you use it next time. That's how the cost remains fixed per sample. So you don't have to have complicated costing systems as well to charge customers or users.
Okay.
Yeah.
All right. Is it really true that you don't have to prepare the consumables with the screen tape anymore?
I would say yes. The only thing you need to do is to put the tape in the machine, make sure that the tips are loaded, and add the loading buffer to the sample. I don't do much lab work anymore, unfortunately, but this is one of the things that I have the chance to do every now and then because it's so easy.
All right. So do you know if the instrument needs any prep or warm-up time?
No, for the same reason. If you decide to run a sample now, just go on. If it's not on, you can switch it on, and it's ready to use instantaneously.
Okay. In order to achieve the same sensitivity as a Bioanalyzer, my current system uses too much of my sample. How much does the TapeStation need?
So routinely we use 1.5 microliters. We had instances where we wanted to try and load less, so it can be we lower it down to 1 microliter for precious samples, in which case you may want to add a bit more buffer or dilute the sample itself in some other ways. So the recommended minimum volume for the instrument to work correctly is 3 microliters. So as long as you are about there, you're fine. I've been playing around with it, and I managed to get good results within 2 microliters, although sometimes misses the sample. So short answer is 1.5 routinely, and 1 can be used with some modifications.
All right, Donna, we're back to you. Someone is wondering, does the TapeStation run a sizing ladder along with the samples or separately in a tape?
So you actually have the option of running a ladder. We provide a standardized software ladder within our software. So if you wanted to use all 16 lanes on our screen tape just for sample, you have the opportunity to do that, and then you can just add in a ladder at the end. You can also use one of the lanes to run a ladder if you so wish. And then you have the option within the software to then save that ladder. So for instance, if you wanted to run maybe 96 samples and you only wanted to run 1 ladder for those 96 samples, you could just run it on the first screen tape, and then you can save that image and apply it to all of the other lanes that come off that same plate.
Thank you, Donna. Our time's up. Thanks for listening in, and thanks for the great questions. I'm glad we could answer so many today. This webinar will be archived for the next 6 months on our website, www.GenEngNews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank our panelists, Paolo Piazza and Donna McDade-Walker, as well as our sponsor, Agilent Technologies. We'll be sending a survey shortly and hope you take the time to respond. Your comments help us continue to provide topical and timely webinars. Thank you.